Okay, first, I'd like to summarize briefly some of the points made in the introduction of this particular article that are related to alertness, particularly in the forebrain, and how alertness and cortical arousal are correlated. So alertness or cortical arousal are mediated by many ascending pathways with neuronal components that project from the upper brainstem, which you can see over to the right, kind of to the bottom of that picture, near the junction of the pons and the midbrain. One of these pathways will innervate the thalamus and the other will innervate the posterior, that should be posterior, hypothalamus and forebrain. Now all of these pathways, or both of these pathways, will include key cell populations that are cholinergic, noradrenergic, serotonergic, dopaminergic, and histaminergic. So if I were a student, I would look up the meaning of these terms and understand which neurotransmitters might be released from these particular cell types and what effects they might have on the sleep-wake cycle. Some key locations of these cell populations that are important for the sleep-wake cycle, particularly here we're talking about alertness, is the pedunculopontine and lateral dorsal tegmental nucleus, which is pointed to by the blue arrow, and they're in orange off to the right. The locus coeruleus, which is pointed to by the beige arrow. The dorsal and the medium raphe nucleus, pointed to by the orange arrow. And the tuberomammillary nucleus, pointed to by the red arrow. So these are key areas in the brain that are important for arousal. I mean, there are more than this, but for now, we'll talk about these. So projections from those cell types discussed in the previous slide fire a characteristic pattern to promote arousal that is actually inhibited every 24 hours or so by neurons of the ventrolateral preoptic nucleus, the VLPO, and you guys know that as the sleep center. Also, they're inhibited by the sleep-active gamma aminobutyric acid, or the GABAergic neurotransmitter signaling mechanisms, or the cholinergic. The interaction between the VLPO and the branches of the ascending arousal pathway is mutually inhibiting, like an on-off switch to enable stable state of wakefulness and sleep. So when people have sleep disorders, it represents the pathology of this switch, causing individuals to suffer from state instability. And here, when we refer to the term state, we mean state of sleep versus state of wake. Sometimes these are referred to as behaviors. So the behavior of being awake, the behavior of being asleep. So what you might be able to recognize, particularly when you're looking at an EEG on an epoch of a sleep study, is wake intruding into sleep. So you might recognize wake and stage 1, wake and stage 1. At the same time, you might recognize sleep intruding into wake, and this is classic of the narcoleptic patient. Now keep in mind our discussion about the RAS, the reticular activating system. And when we talked about this before, we were talking about the thalamus being the gateway into the RAS. And when it's open, then the person can recognize external or environmental stimuli, bring that to the brain, interpret it, and produce some kind of response. The RAS is most important for this. The RAS, the reticular activating system, consists of two branches. And here, branch 1 is in orange, and we talked a little bit about that in the previous slides, containing cholinergic structures of the brainstem and the basal forebrain. Like we mentioned before, it innervates the thalamus, activating relay neurons and activating the reticular nuclei essential for thalamocortical transmission. It also consists of the principal thalamic nuclei, the PPD and the LTD, which you can see in orange, that are going to be most active during wake and REM sleep, slow during non-REM sleep, and when they are high, cortical activity is reduced. Or I'm sorry, in their presence, cortical activity is reduced. Transmission of the reticular nucleus of the thalamus is important as the site functions as that gating mechanism that we talked about before. It blocks the generation of thalamocortical rhythms and promotes a state of excitability and wakefulness. Cortical arousal may also be regulated by projections from the upper brainstem to the midline, and the intralaminar thalamic nuclei include the reticular formation, which is an intermesh neuronal networks throughout the brainstem, the parabrachial nucleus, which is pointed to by the blue line, and the monoamergic system. So from this point on, I'll just call it mono, which is a combination of neurons that have the neuron types discussed in slide 2. The second branch of the RAS, or the reticular activating system, is what we see here in red, and we've talked about a lot. It projects into the lateral hypothalamus, the basal forebrain, the cerebral cortex, and it's comprised of monoaminergic cell populations, which we'll refer to as mono, such as the noradrenergic norepinephrine of the locus and choriolis, the serotonergic dorsal and medium raphe nuclei, the dopaminergic of the ventral periquinoctial gray matter, and the histaminergic of the TMN. These serve broad action potentials that discharge rapidly during wake, but they slow during non-REM and will be slightly, if not maybe not active in REM. There are other cerebrocortical afferents that might play a role in this, such as the lateral hypothalamic peptidergic, which contains melatonin concentrating hormone, highly active during REM, and or orexin or hypercretin, which is low in REM. And the basal forebrain nuclei, which is of course going to produce acetylcholine, which is high in wake and REM. And you can see them abbreviated over in the right, and when you're looking at the article from which this PowerPoint came from, you can see the abbreviations in there. Okay, switching gears to the sleep side, the VLPO, which we talked about a little bit in class. The extended VLPO neurons connect with the pontine, which are cells in the sites in the pons, implicated in REM sleep gating. And these areas are going to include the LPD, the dorsal raphe nucleus, the locus coeruleus. So keep in mind that the VLPO is going to be inhibitory in regards to the areas of the brain that result in wakefulness or alertness. The VLPO cluster provides output to the histaminergic neurons of the TMN also, which are typically active during wake. They are going to reduce firing during REM, and they're going to cease discharge during REM sleep. On the other side of things is the monoaminergic arousal system afferents connected with the VLPO, such as the norepinephrine and serotonin systems. These are released from the locus coeruleus and median raphe nucleus. They inhibit the VLPO, but the VLPO also inhibits them. Also here, on this side of things, is the GABA and galanine neurons produced by, or neurotransmitters produced by the TMN. So if you look at the bottom of the screen, it states the dynamic. The interaction between the VLPO and the monoaminergic system is really what results in this sleep-wake switch. One inhibits, one excites. One inhibits, one excites. So the VLPO inhibits the monoaminergic system to induce sleep. The monoaminergic system inhibits the VLPO to induce wakefulness. Looking at this picture here, the red spot in the middle of the skull represents the relative location of the VLPO, which is important for sleep. So to discuss the interactions between the arousal and sleep areas of the brain is this nice diagram that I got from a different paper. Sometimes when you're reading an article, you might find that the pictures or the diagrams or the writing doesn't really tell you the whole story that you want to know, and you might have to go to other resources to fulfill that requirement, and that's what I did here. So you can see this nice diagram. It actually came from a Nature paper. And if we look at the picture, keep in mind that the solid arrows represent excitatory input. The broken lines represent inhibitory input. Thickness of the arrows and lines will represent the relative strength of excitatory or inhibitory input. So the thicker they are, the more strength there is. And the circle sizes indicate the relative activities of each respective region. So during awake state, the VLPO here in yellow, it's a small circle, so it's not very active. It's a major sleep active center. And you can see over here in brown, the monoamergic neurons really active because the circle is big. And you can see how it inhibits the VLPO. Okay, at the same time, the monoaminergic neurons seem to inhibit the orexin neurons, which actually activate the monoaminergic neurons. So the monoaminergic neurons are responsible for maintaining wakefulness. They send excitatory projections to the thalamus and the cerebral cortex. They send inhibitory projections to the VLPO, and they inhibit those orexin neurons. Now the thing about the orexin neurons is that they excite the monoaminergic neurons, so they will stimulate wake. But at the same time, they will be inhibited by the same system. This provides balance. When we look to the left of that diagram, you can see the limbic system, the SCN, you guys all know what that is, and energy balance, or what I like to call homeostasis, all play a role in how active those orexin neurons will be to activate or stimulate wakefulness. Okay, on the opposite side, the right-hand side, you can see the same diagram with a little bit difference in the size of the relative circles representing sleep state. And here, the VLPO is larger than the other ones because it's highly active. It maintains sleep by inhibiting the monoaminergic neurons and the orexin neurons. Okay, so let's next talk about the brainstem control of state stability, sleep versus wake. And in the first part of this diagram, A, what we see is the orexin or the hypocretin producing neurons active. We see an on arrow going here that promote wakefulness. Remember that the orexin hypocretin neurons are located and therefore produce oxyorexin by the posterior hypothalamus. The receptors are found in major sleep regulating nuclei, like for the oxyorexin 1 receptor, the locus coeruleus, for the oxyorexin 2 receptor, the TMN. Both oxyorexin 1 and oxyorexin 2 receptors are located in the medium raphe nuclei and the mesopontine reticular formation. So up here at the top in part A of this panel, you can see hypocretin or orexin activating the LC, the TPN, and the raphe nuclei, which will produce a state of wake. At the same time, they're inhibiting the VLPO, which will inhibit sleep onset. If you look at panel B, you can see that the VLPO now is active, and it has an inhibitory role on the orexin to inhibit wake arousal. This article also discusses a few sleep disorders, one of them being insomnia, which states that it is pretty common in our population and attributed to stress. So if there's a high stress level, basal stress level relative to norm, then there will be an enhanced hypothalamic function that increases pituitary release of hormones that are important for cortisol secretion. This is going to have a negative effect on metabolism. So it's supposed to be a compensatory response or homeostatic response in which there is stress, we need to have more hormones that increase metabolism for energy. But if this is happening during sleep, we're actually inducing more stress and potentially predisposing a person or ourselves to diseases such as metabolic syndrome, atherosclerosis, diabetes, hypertension, depression, and whatnot. So when somebody has excessive stress that for some reason they're not able to mentally control well, there's going to be an increase in this hypothalamic pituitary adrenal gland axis of function, overall leading to an increase in cortisol and adrenaline, which we call epinephrine. All of these are going to induce sleep disorders, primarily because of sleep fragmentation or prolonged sleep, I should say, wake time. Another disorder discussed in this article is narcolepsy. And what's happening during narcolepsy is that the orexin neurons are either not functioning or for some reason there is a decreased level of orexin or hypocretin hormone being released. And when that's happening, we fail to activate the monoaminergic neurons normally. When that occurs, the monoaminergic neurons and the VLPO neurons set up a mutually inhibitory circuit. This causes unwanted and abrupt transitions between the states, sleep state versus wake state. Activity in one of the competing sides shuts down inhibitory inputs from the other side and therefore disinhibits its own action. If either side begins to overcome the other, the switch abruptly turns into the alternative state. And most commonly what you see in narcolepsy is there will be an abrupt turn from wake to sleep state. So make sure you read about narcoleptic patients going into REM very quickly or having the state of cataplexy. The article also discusses fragmented sleep and the role that plays in poor wake sleep switch function. This is an epic from a polysomnography that demonstrates obstructive sleep apnea. So if you look to the left and kind of browse down that list of terms, you can see NAF. NAF represents nasal airflow. Right under that is THO, which represents thoracic movement. And under that, you can see ABD, which represents abdominal movement. So you can see the nasal airflow about, let's see, maybe that would be, if you look at 30, and then you go backwards about 3 of those points up to the bottom, and you can see how the airflow starts to go down to nothing. But at the same time, we still have thoracic and abdominal movement. This represents obstructive apnea. At the same time, if you look in the EEG, which is represented by C3A2 all the way to C401, you can recognize that there's an increase in amplitude and frequency about 2/3 through the obstructive apnea. This represents an arousal in the brain. So even if the patient doesn't completely wake up, there is a brain arousal that results in fragmented sleep and can cause metabolic disorders because of the stress response. The article also talks about restless leg syndrome. And there are many reasons for restless leg syndrome, but it's important to note that it's different than periodic leg movements in that there's not a spontaneous movement. The patient wakes up because they feel a tingly or a creepy crawly sensation in their periphery, particularly in their legs, that makes them want to move. So it wakes them up. And most commonly, this is caused by a problem with iron storage. So panel D over here, physiological state, there's two genes at the top, two proteins, colon 3 and BTBD9, that when they associate together play a role in facilitating a downstream cell signaling pathway that results in normal iron storage. This also increases or facilitates normal dopamine levels, which both are important for normal motor drive and function. If these levels are normal, physiological, then the patient will not experience RLS and they will maintain a good sleep pattern. However, if for some reason this colon 3 protein and BTBD9 cannot associate and therefore do their function, this process that's important for maintaining normal iron storage and normal dopamine levels is altered. This makes the sensory motor drive hyperactive, causing this tingling sensation in the legs that is going to wake up the patient and cause sleep fragmentation. Okay, and then finally I found this diagram that pretty much tries to display the potential disorders, neurological disorders, and their effects on sleep and other problems. So if we look at dementia over here on the left side towards the middle, it discusses how there might be a circadian rhythm related sleep disorder, and we've talked about that in class. Dementia also facilitates nightmares and hallucinations and delusions and dyskinesia, all of which can cause sleep fragmentation. On the top, it says disease-related change. Some diseases can cause a disturbance of the sleep-wake cycle, alterations in the non-REM and REM sleep switch, and their arousal system. All of these types of things can actually cause motor neuron problems that stimulate dyskinesia. And of course, there's drugs or particular habits that might induce fragmented sleep. Daily habit is very important, and you will probably talk about sleep hygiene with Abdullah. If you guys don't, let me know, and we'll talk about it in class a little bit. So just think about this figure and what it might mean in regards to neurological causes of sleep fragmentation and how that might be translated into your patient assessment. Another thing I want you guys to think about is most often respiratory therapists think that polysomnography is boring because they basically just think of it as assessing for obstructive sleep apnea. But as you can see from this particular presentation, there are many different neurological and peripherally physiological problems that can cause sleep disorders. So it's really a study of the brain and how it responds to its environment and the peripheral nervous system. And that's it. I look forward to hearing your presentations and talk to you soon.